Hi, I'm Dr. Jim Zoval, a professor of chemistry at Saddleback College. You're about to watch one of my video lectures. If you like this lecture, you can get the entire Allied Health General Organic and Biochemistry course from the website address you see on the screen right now. Be sure to type the underscore between VOD and pages in this address. Enjoy the video. You don't need to know 14 to understand 15. 15 you need to know up to chapter 13, but get started on that right away. Let's start not looking at the polymers, the DNA and RNA. Let's look at the building blocks, the molecules that make those polymers, the residues. These polymers, nucleic acid polymers, consist of molecules called nucleotides. Just like polysaccharides were made from monosaccharides, proteins were made from amino acids. Nucleic acids are made from nucleotides. There's 3 parts to a nucleotide. This is a small section of a DNA molecule. It has 1, 2, 3, 4 nucleotides. Let's consider the 3 parts of each of these 4 nucleotides. There is a phosphoric acid part. That's called phosphoric acid part because it comes from phosphoric acid. There's a monosaccharide. Look familiar? And then in blue, in DNA and RNA, there'll be one of 4 organic bases. Remember, for amino acids, we saw 20 different side chains. These are analogous to the side chains. Whether it's DNA or RNA, you'll have one of 4 different bases. Let's take a look at each one of those parts. So the one part that's called the phosphoric acid part, that comes from phosphoric acid. Phosphoric acid has 3 hydrogens that could come off. And so depending on what the pH is, it'll exist in one of these different forms, just like we saw for amino acids and any other acid, right? Acid forms, base forms. But here in low pH, very, very acidic solution, we got 3 hydrogens. As the pH increases, you have less and less hydrogens present. Here are the 2 forms that are present at physiological pH. The pKa of this is right around 7, so you have about as much of this one as you do of this one, about equal amounts at physiological pH. The second part is the sugar, the monosaccharide. All nucleic acids, whether it's DNA or RNA, contain either this sugar or this sugar. One of those 2 monosaccharides. It's based on ribose. Ribose is a 5-carbon sugar. Here's carbon 1, 2, 3, 4, carbon 5 up here. Notice we have deoxy right here because on carbon number 2, deoxy, it is missing an oxygen. It doesn't have the OH there, as does ribose. And furanose, that's what we call 5-member sugar rings, furanose. And the third part of a nucleotide is the organic base. These organic bases in DNA will be one of these 4, thymine, guanine, cytosine, and adenine. In RNA, you don't have thymine, you have something called uracil. That's coming up later in your notes. We could divide these organic bases up into 2 categories based on how many ring structures there are in them. This is a molecule called purine. Notice there's 2 ring structures, a 6-member and a 5-member. So the organic bases with this basic ring structure, a 6 and a 5, are called purines. There's 2 of them, adenine and guanine. The other category are called pyrimidines. Notice this is the pyrimidine molecule. It's a 6-member ring with 2 nitrogens in it. All these have that 6-member ring with the 2 nitrogens in it. Now they have other things, and that's why this is, for example, cytosine. It has an amino group here and a carbonyl group here, where thymine has 2 carbonyl groups. Uracil has 2 carbonyl groups, but it doesn't have that methyl. It doesn't have a methyl here, where thymine has the methyl there. If we start building these from their components, from the phosphate, the sugar, and the organic base, we'll see that when you combine the sugar, whether it's the ribose or the deoxyribose, with the organic base, you get what's called a nucleoside. We're not to nucleotide yet. Once we have the phosphate, then we got the nucleotide. With just the sugar and the organic base, we call that a nucleoside, S-I-D-E. So if you start with ribose as the sugar, and you add one of those organic bases, you get ribonucleosides. When the organic base is adenine, we have adenosine, ribonucleoside. When the organic base is guanine, we have guanosine. Likewise, cytidine and uridine. If we start with the deoxy, we get deoxyribonucleosides, and we'll get the 4 deoxyribonucleosides. Just want to remind you guys the difference between oxy. If you look on carbon 2, you have an oxygen there. Deoxy, you don't have that oxygen there. Why does it say 2'? We have this double ring system here and the single ring system here. There's a priority. Which ones get numbered with their carbons as 1, 2, 3, 4, 5. Right? Notice up here 1, 2, 3, 4, 5. So we had to distinguish these carbons in this ring system here. So that's 1', 2', 3'. I don't expect you guys to know that priority system. Just know that when we're talking about the carbons in the sugar ring, we use prime on them. Notice the organic base is connected to the anomeric carbon. So when you have the organic group in the sugar, you have a nucleoside. When we add the phosphate, then we get the nucleotide with the T. Notice the phosphate got added to carbon 3 right there to make a phosphoester bond. We've seen phosphoester bonds. We saw that when we looked at phospholipids. I want to remind you guys about that. If we react a carboxylic acid and an alcohol, we get an ester. That's the esterification reaction, right? Notice this is kind of like a carboxyl group. If that was a C, you'd have the double bond O and then an O right there, right? It's kind of like a carboxyl group. So when we do this with a phosphorus, we get a phosphoester. Let me show you the bonding and how that happens in this case. So I'm going to draw the sugar. On the anomeric carbon, we have a bond to the nitrogen in, I'm just going to draw this and call this the organic base. We saw the different organic bases. And now I'm going to draw the phosphate. I haven't made the phosphoester bond yet. I'm going to draw this phosphate as it would exist at physiological pH. It has 1 hydrogen on it, sometimes 2. Sometimes you'd have another hydrogen on one of these. It doesn't matter for this example. And I'm going to make the phosphoester bond. Notice if that was a carbon, this would look like a carboxylic acid, right? That carbon double bonded oxygen, OH. If we did a reaction with an alcohol, we'd get an ester. Look at this is like an alcohol, an ROH. We don't have a carbon there, we have a phosphorus, but we're going to make the same type of bond. What we're going to do is we're going to take this OH and that H, and we're going to make a phosphoester bond. That's what we have here. Let's look at this other bond here. Notice N-glycosidic bond. So in the nucleotide, the sugar and the organic base residues are connected by an N-glycosidic bond. We've seen glycosidic bonds from one sugar to another sugar before. Here we go from sugar to the organic base, to the nitrogen on the organic base. That's why we call that an N-glycosidic bond. Let's take a look at how that bond is formed. Somewhere in all these different organic bases, there'll be a nitrogen with a hydrogen on it in the organic base. So this is the rest of the organic base. And I have the beta form of the sugar. That's what happens in nature. It reacts with the beta form. Why is it beta? It's because enzymes catalyze the production and formation of these bonds, and it does so with the beta. What I do is I take one of the hydrogens off the organic base and that OH to make a water molecule, and I make the N glycosidic bond from the sugar to the organic base. This will always be from the anomeric carbon to one of the nitrogens on the organic base. Now, you don't need to memorize which one of the nitrogens on the organic base will react, because it's beyond the scope of this class for me to have you guys memorize too much. And this exam is open book, open notes, so you'll be able to look and figure it out. So we saw a couple fairly new bonds here. We saw the phosphoester bond. You guys have seen that once before. And now we see an N-glycosidic bond. You saw a glycosidic bond, but you haven't seen an N-glycosidic bond. When you do this, when you make the organic base bond to the sugar and to the phosphate, you have a nucleotide. Those are the basic building blocks of nucleic acids. In the example I did, and in this example, notice the phosphate bound to carbon number 3 in the sugar ring, the 3' carbon. We call that a 3' nucleotide. That's not always the case. Also in nature, we see binding to carbon number 5. Ribose is a 5-carbon sugar, so that's carbon number 5. We could see 5' nucleotides. For example here, if the phosphate is bound to carbon number 5, it's a 5' nucleotide. If it's bound to carbon number 3, it's a 3' nucleotide. We see both of those in nature. If you start with a ribonucleoside, the organic base in the sugar, nucleoside, ribose, it's not deoxy, it's ribose, and you add that phosphate, you get a ribonucleotide. If you start with a deoxyribonucleoside, the sugar, deoxy sugar and the base, and then you bind a phosphate, you get a deoxyribonucleotide. You guys try one. I want you to bind these 3 together to make, remember, there's 3' and 5'. I want you guys to make the 3' ribonucleotide out of that. If you think you know what to do, go ahead and start. If you want to hit, pay attention. What you're going to do is you're going to bind the organic base to the anomeric carbon through an N-glycosidic bond. You're going to take this OH, and it's this nitrogen that it binds to. You wouldn't have to know that for the test. You'd have open book and open nose. So you're going to take this OH and that hydrogen, make a water molecule, you don't have to draw that, but then make a bond between the anomeric carbon and that nitrogen. That gives you the ribonucleoside, and then to make the ribonucleotide, the 3' ribonucleotide, you're going to bind the phosphate to carbon number 3. You're going to make the phosphoester bond there. Check your answer. Here it is. Now you may have drawn this kind of, the bond's still to that nitrogen, but you may have drawn it over this way. That's fine if you have this, remember, because that's a single bond. This thing rotates around. You may have drawn it like that. This whole thing flipped over there. That's fine. Don't forget to put the formal charges on the oxygens with just one single bond going to them. Hi, I'm Dr. Jim Zoval. If you like this video and you like to get the entire course or just some of the chapters on video, I have the entire course and video lectures at the website you see on the screen right now. They're available on demand. The period of the rental is for a full year, so you'll have this video through the entire course. You could watch it as many times as you want, whenever you want. Happy studies.